Well, welcome everyone to this podcast on the Calvin cycle and its consequences, part of BIocore 383. So far, you'll recall that we've said that the reactions of photosynthesis can be grouped into two sets of reactions. First, the light reactions, which involve an input of light and water and result in an output of molecular oxygen and compounds that will be used in the second phase of photosynthesis, the Calvin cycle. Those two sets of products are ATP and reducing power in the form of NADPH. The ATP and the NADPH then are used internally in the chloroplast to fuel the carbon fixation reactions of the Calvin cycle. So in the case of the Calvin cycle, the input is going to be gaseous CO2, carbon dioxide, which is going to be fixed in a cyclical set of reactions, ultimately producing 3 carbon molecules for many plants, or trioses, and ultimately these trioses will be converted to 6 carbon molecules in the form of things like glucose. So at the end of the day, we input carbon dioxide and we output sugar. So let's talk about these second sets of reactions in the Calvin cycle. And what we want to do here is to first focus on where these processes are occurring, how the outputs of the cycle get to regions outside of the chloroplast. Then we want to talk a bit about the logic of the Calvin cycle, just as we did for the TCA cycle. We'll talk a bit about the details of the Calvin cycle itself. And then we'll talk about some consequences of the Calvin cycle that result, at least in part, due to the inefficiency of one of the key enzymes that catalyzes one of the most important reactions of the Calvin cycle, rubisco. Right, so I told you already that the Calvin cycle reactions occur in the chloroplast, and that's true. So step 1 here in this slide shows that the Calvin cycle reactions are occurring inside the chloroplast, using ATP inside the chloroplast from the light reactions, as well as NADPH from the light reactions. And then gaseous CO2 diffuses across the double membrane on the outside of the chloroplast and is used by the Calvin cycle in carbon fixation. This all happens in the yellow colored part of this diagram, the stroma. Eventually, the 3 carbon molecules, which are actually phosphorylated, so they are triose phosphate, shown by the green box in this slide. Ultimately, they can either be converted into a storage form of sugar that we've seen that plants use called starch. That happens in the stroma. Or those triose phosphates can be transported outside of the chloroplast into the cytosol, and they can be used to make sucrose. Now, the Calvin cycle itself is named after one of its discoverers. Often it's called the Calvin-Benson cycle, but I'll call it the Calvin cycle most of the time in the rest of this podcast. So I actually did my PhD at UC Berkeley, and there's a building there called the Melvin Calvin Lab. Calvin did a lot of his work at Berkeley. He was really a brilliant biochemist. And his key approach was to use C14 labeled carbon dioxide, radioactive carbon dioxide that could be traced through the reactions which now bear his name. And what he found was that after you introduce radioactive CO2 to plants, they rapidly incorporate those into 3 carbon molecules, trioses, and only later do they produce sugars. This is modified from figure 1111 from World of the Cell and shows an overview of the Calvin-Benson cycle. As we saw with the TCA cycle, carbons are represented by little gray balls. Phosphates you see by the letter P inside of a yellow circle. So what we want to do is to look at the overall logic of the Calvin cycle here using this slide. So the first step in the Calvin cycle is to take gaseous CO2. Actually, we take 3 of these, and we are going to incorporate them into a set of 3 existing molecules called ribulose bisphosphate. And the result is going to be that we will produce 6 molecules that have 3 carbons each, and these are molecules of 3-phosphoglycerate. Once we have these three phosphoglycerates or phosphoglyceric acid, PGA molecules produced, then we use the ATP that we generated in the light reactions, as well as some of the reducing power in the form of NADPH, to carry out a series of reactions. And the ultimate effect of this is to produce 3 carbon molecules, or trioses. We produce 6 of these, and these 6 trioses have an interesting fate. So only one of these is used for synthesis of other organic molecules, like sugars, for example, or starch. 5 out of the 6 trioses actually continue within the cycle. There are some dephosphorylation reactions, then ATP is used, and ultimately, as a result of these reactions, we regenerate ribulose bisphosphate. So we have carbon fixation at first, then we have some reducing reactions which involve NADPH. ATP is used at a couple of places in the cycle, and ultimately we regenerate ribulose bisphosphate, which can then enter another round of the Calvin cycle. So you see then that it's here that the carbon is incorporated through carbon dioxide that was part of the balanced equation of photosynthesis that we started our discussion of photosynthesis with. All right, so in the end, if you look at this, what you'll see is that for each CO2 we need 3 ATP and 2 NADPH. So that's an overview of the Calvin cycle, and it's worth pointing out that the enzyme that carries out the initial carbon fixation is very important. It's been estimated that it's the most abundant enzyme on the planet, and it's called ribulose bisphosphate carboxylase oxygenase, or because that's a whopping mouthful, it's often abbreviated Rubisco for short. Rubisco is an interesting enzyme. It is extremely inefficient. It fixes only 3 carbon dioxide molecules per second, and if you compare that to typical enzymes, often they'll be catalyzing reactions at 1,000 molecules per second of their substrates. So you need a lot of Rubisco to carry out these reactions because of its inefficiency. Secondly, a key thing that plants ultimately have gone to great pains to deal with is that not only is it slow at doing its job, it actually has trouble distinguishing carbon dioxide as a substrate from molecular oxygen. And that's why in its name it has the words carboxylase and oxygenase. It can carry out either reaction depending on the partial pressure of CO2 and O2. And so it turns out that this means that this enzyme actually doesn't catalyze the reaction we want as part of the Calvin cycle much of the time, and the plants have to deal with a complicated series of recycling reactions to deal with that inefficiency. So Rubisco is surprisingly inefficient, and it has to be said that attempts have been made to genetically engineer Rubisco to make it more efficient, and thus far all of those attempts have failed. So there's something about this enzyme and its normal intended reaction, if you will, that has joined it at the hip with this unintended reaction, and so plants have to deal with this. All right, so the Calvin cycle reactions occur, just as a reminder, within the stroma, and we said that the light reactions which occur in the thylakoid membrane generate NADPH in the form of reducing power, and they generate ATP through chemiosmosis. So let's go back then and look in more detail at the Calvin cycle and some of its reactions. And to do that, we'll look at figure 1112 from your text. So remember, the first step of the Calvin cycle is to take 3 molecules of carbon dioxide. To balance this reaction, we need 3 molecules of water as well as an input. And we have 3 ribulose 1,5-bisphosphate molecules that have been moving around the cycle. And we combine these to generate 6 molecules of 3-phosphoglycerate or 3-phosphoglyceric acid. In many plants, this is the first step of the Calvin cycle, and we call this carbon fixation because the carbon dioxide from the atmosphere is getting incorporated or fixed into these 3 PGA molecules. Right, so there are 6 3-phosphoglycerate molecules. Then we use some ATP to generate 6 molecules of glycerate 1,3-bisphosphate. And then those 6 molecules of glycerate 1,3-bisphosphate undergo reduction, and this is made possible by the reducing power NADPH that we produced in the light reactions. So we need 6 molecules of NADPH to reduce these 6 molecules of glycerate 1,3-bisphosphate. So we do that, and this generates 6 molecules of glyceraldehyde 3-phosphate. So these 3 molecules of glyceraldehyde, or these 6 molecules of glyceraldehyde 3-phosphate, then, as we said, have different fates. Only one of these can be used for biosynthesis. 5 out of every 6 molecules continues around the cycle. So we lost a phosphate, which you can see by the boxed hydrogen there. There was a phosphate there that's been removed. And again, only 1 out of 6 of these 6 glyceraldehyde 3-phosphates are used to make biomolecules. The remaining 5 continue around the cycle, as we've said. And what they do then is to regenerate the molecules of ribulose 1,5-bisphosphate, sorry, that's a typo in the slide, that are utilized then for another round of the Calvin cycle. So that's in detail what's going on with the Calvin cycle. So you see, if you look at these reactions of the Calvin cycle, that you actually need NADPH in the form of reducing power and you need ATP. But it turns out that the overall utilization of reducing power and ATP differs at different times of the day for plants. Different plants have different requirements for ATP versus NADPH. And so plants are pretty good at fine-tuning how much ATP versus reducing power they produce through the light reactions, which is where those molecules came from. And it turns out that plants can engage in something called cyclic electron flow. So you remember that photosystem 1, it's actually the second of the 2 photosystems in the light reactions of photosynthesis, absorbs a photon, and it's excited to a state in which it likes to give up that electron to electron transport chain molecules. And in cyclical electron flow, this also occurs. However, in cyclical electron flow, instead of going all the way to the production of NADPH, if we don't need that much reducing power, and instead we'd rather make more ATP, then cyclical electron flow forestalls the generation of NADPH, and instead we kick those electrons back through a cyclical electron flow process to produce more protons, which can be used to make more ATP. So we go all the way through to ferredoxin, and the result is that we release more protons instead of making reducing power by not going on to make NADPH. So plants can fine-tune ATP generation versus NADPH generation. That's an interesting feature of photosynthesis. So depending on the needs related to the Calvin cycle, they can do this process. And you'll remember that both of these are needed for the Calvin cycle, and so this is a nice adaptation to optimize the reactions of the Calvin cycle. Okay, so the Calvin cycle produces these 3 carbon molecules in many plants called trioses, and let's see what happens to them and how plants utilize those trioses. So this is figure 1114 from your text. And what this shows is that we can take glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and we can actually use these 3 carbon molecules to produce various kinds of energy-containing compounds. If those trioses remain in the stroma, then they can go on through fructose 6-phosphate to make glucose 6-phosphate. Ultimately, there's an isomerization to make glucose 1-phosphate. Some ATP can be used, and this can be polymerized ultimately and added on to a starch molecule. That's on the right in the stroma. For all of this to occur, we need inorganic phosphate, and it turns out that the most abundant protein in the chloroplast is a phosphate translocator, and this is an antiporter that catalyzes the exchange of triose phosphates in the stroma on the right in the yellow for inorganic phosphate from the left in the orange from the cytosol. And so this is actually moving trioses and inorganic phosphate between the two compartments, and ultimately then trioses can wind up in the cytosol as well. So in the stroma, the inorganic phosphate is used with the trioses that remain there to make starch. But in the cytosol, these trioses have a slightly different pathway. The initial reactions are the same. Ultimately, you get to glucose 1-phosphate, but then a cousin of ATP, UTP, is actually used to make something called UDP glucose. This then is glued on to a fructose 6-phosphate molecule to make sucrose 6-phosphate. Ultimately, that's dephosphorylated, and we get sucrose, which, as you know, is the component that is familiar to us as table sugar. So these trioses can be made into disaccharides in the form of sucrose in the cytosol, but in the stroma, they're made into starch. All right, so at this point, it's really useful, I think, to take a step back and compare what's going on in the mitochondrion with what's going on in the chloroplast. So you remember in the mitochondrion, we had fats and carbohydrates entering the citric acid cycle at various steps. That's on the lower left. And we also had glycolysis as an input, adding in to the TCA cycle at the point where acetyl coenzyme A comes into the cycle. Now, the goal of the TCA or citric acid cycle is to generate reducing power in the form of NADH, and this is used in electron transport to produce a proton gradient, which ultimately can be used to drive ATP production. The citric acid cycle generates CO2, and oxygen is used as a final electron acceptor in electron transport in the mitochondria. Both use chemiosmosis to produce ATP. Now, what about the chloroplast? Well, in the chloroplast, we have light causing excitation of a chlorophyll at the center of the photosystem, making it very likely that it will give off an electron and generate a flow of electrons through electron transport. In this case, water is actually split, so the opposite reaction, to make molecular oxygen. Those electrons are used in a very similar way, however, to produce a proton gradient, which is going to be used for chemiosmosis to make ATP. There's a second photosystem, photosystem 1, where another photon is absorbed. This allows further electron transport and ultimately allows for the production of another reduced molecule, NADPH, a cousin of NADH. This is then used in a cyclical set of reactions, the Calvin cycle, where CO2 is now incorporated into the molecules associated with the cycle, and the ultimate goal is to make carbohydrate molecules. So you can see there are similarities and yet differences. And I hope you'll think about that a bit more as you continue to study both oxidative phosphorylation in the mitochondrion and the light reactions and the Calvin cycle in the chloroplast. All right, so there's a little bit of unfinished business that we need to discuss, and these are really consequences of the inefficiency of one of the enzymes in the Calvin cycle. So let's come back to that. That enzyme, remember, is called rubisco. And remember that rubisco has oxygenase activity. So rubisco's oxygenase activity yields one molecule of 3-phosphoglycolate and a 2-carbon compound called phosphoglycolate. So you can see there are these two compounds, and these are produced as a result of the oxygenase activity of rubisco. Remember, it's normally the other enzymatic activity of rubisco that fixes carbon into molecules during the Calvin cycle. So this is kind of an unintended side effect of rubisco's lack of specificity in terms of its substrates. So you've got these molecules that come out of the oxygenase activity of rubisco, and depending on the plant, you get differing levels of production of phosphoglycolate and 3-phosphoglycerate. And so we have to deal with both of these using a new set of chemical reactions that we haven't discussed so far. And the ability of plants to deal with this oxygenase activity involves something called the glycolate pathway. So that's shown here. This is figure 1115 from your text. So there's a chloroplast here on the left in yellow, and you can see the grana stacks inside the chloroplast. Remember, the Calvin cycle reactions are occurring in the stroma, the yellow region. But you can see there that oxygen is around, and depending on how much oxygen is present, you'll get more or less phosphoglycolate produced. So that phosphoglycolate actually has to be dealt with in a different organelle entirely. And you can see that the organelle that helps to deal with it is the peroxisome. Now you'll remember that peroxisomes deal with all kinds of things, and in particular they deal with various oxygen species, including hydrogen peroxide and a number of things. And the glycolate is dealt with in the peroxisome. You can see that there are a series of reactions, and they actually involve additional oxygen coming in. The first of these reactions produces hydrogen peroxide, and that's why it's good that these reactions occur in the peroxisome, because peroxisomes are uniquely equipped to deal with hydrogen peroxide and similar species. So that glycolate is transformed into something called glyoxylate, and then glutamate, an amino acid, is used to produce another amino acid from glyoxylate, the amino acid glycine. Glycine is an amino acid. The cells of a plant know how to deal with that. And in particular, that glycine can diffuse into the mitochondrion. And mitochondria actually can deal with the glycine by using some reducing power, or by generating reducing power in the form of NADH, I should say, and converting that glycine to serine. And then it's been determined that that serine can diffuse back into the peroxisome, as you see on the bottom part of this slide. That serine is then transformed into something called hydroxypyruvate, and then we actually use up a molecule of NADH to produce glycerate. And that glycerate can re-enter the Calvin cycle as 3-phosphoglycerate. So that oxygenase activity produced 3-phosphoglycerate. That was okay, but the phosphoglycolate had to be dealt with by moving it to the peroxisome, transforming it through a series of reactions to an amino acid glycine, moving it to the mitochondrion, where it's then converted to serine. We actually make a molecule of NADH. The peroxisome actually uses a molecule of NADH at the end of the day to convert that serine to glycerate, which can go back into the chloroplast. So you see we have a 3-organelle recycling system, all as a consequence of the tremendous inefficiency of the Rubisco enzyme. All right, so it turns out that peroxisomes, as I said, are a good place for this. So remember that hydrogen peroxide in the peroxisome actually can be dealt with through a molecule called catalase. This actually liberates oxygen, well, half as much oxygen as was used in the initial reaction, and generates some water. So that's good. And then ultimately that oxygen, if you look at the total net usage of oxygen here, we're actually using a bit of oxygen. So because we're using oxygen, biochemists often call this process photorespiration. And photorespiration then is summarized here. So we took 2 phosphoglycolates, which were due to the oxygenase activity of Rubisco. Each of those has 4 carbons. We lose one of those carbons as CO2 if you look at all of the reactions here, and that happens in the mitochondrion. But we've retained 3 of those carbons as 3-phosphoglycerate at the end of the day back in the chloroplast. So of the 4 carbons that we lost through this oxygenase activity of rubisco, we actually get 3 of those back. So this is a 75% efficient recycling mechanism. Compared to industrial recycling mechanisms, this is very, very efficient. All right, so there's one other plant physiological issue that we'd like to tackle in this podcast, and that has to do with the way that various plants, depending on their lifestyles, use 3 or 4 carbon molecules. So we've been talking about triose as an output of the Calvin cycle. That's a 3 carbon molecule. And many plants, called C3 plants, fix carbon into 3-phosphoglycerate. That, again, has 3 carbons, as I just mentioned. And if you look at the internal structure of the leaves of a C3 plant, they look like this. So there are cells called mesophyll cells that are colored green in this diagram, which is figure 1116 from your text. And what this shows is that they are the engines of photosynthesis in a C3 plant. You remember from BIocore 381 that plants can respire, and they do this through specialized structures called stomata. A stoma is singular, has guard cells that can open and close and allow gases to enter the interior of the leaf, or they can be closed to prevent loss of water and other gases. So C3 plants do their photosynthesis in these mesophyll cells. And C3 plants are typically plants that are not dealing with water loss issues. So these are plants where water is plentiful. It's really not a problem for them to be able to respire frequently. But C4 plants are a bit different. They're shown on the next slide. Again, this is figure 1116 from your text. So C4 plants are different. C4 plants often live in environments where water loss is a real problem, potential problem. So C4 plants often close their stomata at various times during the day, during the hottest part of the day, for example, to prevent water loss. Now, if you close your stomata, you have a problem. Remember that the Calvin cycle reactions depend on CO2 as an input. So you've got to fix carbon through CO2. But remember, we had this rubisco enzyme. It's very sensitive to how much CO2 is around versus how much molecular oxygen is around. So if you don't have enough CO2, if your CO2 levels drop because you've closed your stomata, that's going to lead to more phosphoglycolate. You're going to have to run that glycolate pathway to try to recycle things. And so these plants actually use a riff on the Calvin cycle that we've discussed so far. They actually fix carbon not into 3 carbon compounds, but instead into 4 carbon compounds. So that's why these plants are called C4 plants, because they do their carbon fixation with 4 carbon molecules. And let's see how they do that. This is a very simplified view of the C4 pathway. And what you can see in this slide here is that C4 plants actually have 2 sets of cells. They have cells called bundle sheath cells, and they have the cells we mentioned in C3 plants, mesophyll cells. So the difference with C4 plants is that they actually separate carbon fixation, which occurs in the mesophyll cells, by an auxiliary set of reactions from the Calvin cycle. And the Calvin cycle actually occurs in the adjacent cells, in the bundle sheath cells. So again, we have to do this because rubisco is more efficient at catalyzing 3-phosphoglycerate when carbon dioxide levels are high. When they're low, we would get a lot of unintended products. So a different enzyme, PEP carboxylase, is used to fix carbon in the mesophyll cells in C4 plants. And this fixes carbon dioxide into a molecule you may recognize. That we actually mentioned it in the TCA cycle. It's called oxaloacetate. That's a 4 carbon containing compound, as is malate, into which it's converted. Malate then actually moves from the mesophyll cell to the bundle sheath cell. So these 4 carbon compounds are transferred to the bundle sheath cells, and there additional reactions convert the malate to pyruvate. That's a 3 carbon containing compound. and the other carbon is actually released as CO2. This is internal CO2 kind of in the interior of the leaf cell, and that internal CO2 can be incorporated into the Calvin cycle in the bundle sheath cell. And then all of the reactions that we've discussed with the Calvin cycle can occur to generate sugar, which can be transported to vascular tissues and transplanted or transported throughout the rest of the plant. So the C4 pathway is an adaptation, and this all goes back to the fact that rubisco has this oxygenase activity. Now there's another kind of pathway that I don't really want to talk about very much, but it's an alternative to C4, the C4 pathway. And actually different plants in a very similar environment can use this. So sugarcane, for example, in Hawaii uses the C4 pathway. There's another pathway, which is called the crassulacean acid metabolism pathway, or the CAM pathway, and pineapples in the very same environment use that pathway. So let's compare these. This is another way to deal with drought tolerance. All right, so the C4 plants do the spatial separation of carbon fixation in the mesophyll cells into 4 carbon compounds, conversion of the 4 carbon compound into a 3 carbon compound in the bundle sheath cell. The CO2 released as a result of that reaction can enter the Calvin cycle. That's what we've already talked about. So that represents a spatial separation of carbon fixation from the Calvin cycle. CAM plants do something different. CAM plants do it all in the same place, but they do these 2 phases of the reaction at different times. And so in CAM plants, we actually fix CO2 at night into organic acids, which are stored in specialized vacuoles. And then during the day, those organic acids are used to liberate CO2, which can then enter the Calvin cycle. So in these plants, the carbon fixation happens during one phase of the day. The CO2 is released during a different phase of the day and used in the Calvin cycle to make sugar. So CAM plants, it turns out, are very, very efficient at carbon assimilation. So if you compare CAM plants to those that do normal C3 kind of photosynthesis, CAM plants can assimilate over 25 times as much carbon as a C3 plant for every unit of water transpired. So they're very efficient. And we can see that these modifications to the basic Calvin cycle and carbon fixation that occurs during that cycle allow all of this to be possible. Well, I hope this discussion of the Calvin cycle and its consequences has been useful to you. Thanks for listening. We'll see you again soon.